Hi, everybody, and welcome back to Bio 229 at HCC, Holyoke Community College. This is microbiology. I am your host and Professor Mary Annarella, so welcome back. We are finishing up cellular respiration, and I have to tell you, this has been quite a doozy of a lecture. This is Friday night now. I wanted to have the lecture up for you on Friday night, and so I recorded it Friday afternoon, right after lunch, and somehow managed to screw up things so that I accidentally, without realizing it, pressed the button to stop the recording. And so I gave you the most fabulous, amazing lecture on aerobic cellular respiration and photosynthesis, the best ever, shouty caps there, and did not record a single bit of it, and I did not have the intestinal fortitude to re-record it right away. I had to go and recover, and there may have been chocolate involved in that recovery, chocolate and a bit of wine. So this is Friday night, and so I have a bit of wine right here with me as I lecture today. So I suppose we could call this drunk biology. I can't spell drunk, can I? Dun dun dun. I've tried to sell this idea to Nat Geo. They wouldn't go for it, but I think drunk biology would be awesome. Anywho, we're going to finish up aerobic cellular. We have finished up aerobic cellular respiration, but I want to just give you one last little tidbit here. Whereas, okay, if we're in eukaryotic cells and we're talking about the mitochondria, we've got some proteins of the electron transport system in here. Oh, here's ATP synthase. Let's put it in there. A few more. Oh, and just to put it out there that this group here is not the only bit that's there. This entire inner membrane of the mitochondria is just filled with these electron transport protein complexes just embedded in there. So they're in there, and they're all over the place. And that's what's really cool about the inner membrane of the mitochondria, having all those folds. It's an increase in surface area, so you can fit more proteins in there. Anywho, you know the story about how NADH rips those hydrogens out of glucose. And once it gets to the electron transport system, it's like, here, electron transport system, take those 2 electrons and that proton. The electrons get, played hot potato with, and the protons get pumped across the inner membrane of the mitochondria. There are 10 of these things, so, we get 10 protons pumped up here. I'm not going to put them in there, all 10, whatever. You've seen this before. Basically sets up an electrochemical gradient that isn't stable. And anyhow, we've got a high concentration of protons in this intermembrane space, and a low concentration in the matrix of the mitochondria. That's not stable. So a bunch of these protons need to flow through and equalize the gradient, and energy is released when those protons equalize the gradient through ATP synthase. And that gives ATP synthase the energy to take that initial phosphate group and put it back onto ADP to form ATP. That is called chemiosmosis, and this is oxidative phosphorylation. But when enough of these protons flow through, that gradient's going to be equalized, and then no more are going to flow, because we'll have 5 and 5. There we go. So no more are going to flow because it's equalized. But oxygen solves that problem by saying, hey, I'll bind to these 2 electrons and a couple of protons. And it's actually 4 and 4. And form H2O, water. So because oxygen is the final electron acceptor, we call it aerobic if it's O2. there are organisms that don't use oxygen as their final electron acceptor. There are some organisms that don't use oxygen at all. They will use something like, for example, S2, and you'll end up getting H2S. Or nitrate, and you'll get nitric acid, for example. So I just want to put it out there that there are different ways to make ATP. Let's get a slightly different color here. Let's say, okay, respiration. Respiration means you have a proton motive force. And at the end of that proton motive force, to keep that gradient going, you need a final electron acceptor. If that final electron acceptor is oxygen, that is aerobic respiration. Oh, let me qualify this. You need a final electron acceptor that's inorganic. Inorganic. Inorganic means you don't have both carbon and hydrogen. If oxygen is that inorganic final electron acceptor, it's aerobic respiration. And I should have put quotations there. If it's S2 or nitrate, it's or something that's not oxygen, it is anaerobic respiration. Now, sometimes I hear people call anaerobic respiration the same thing as fermentation, and that is so not true. Fermentation involves no PMF. There is no proton motive force, no membrane, no chemiosmosis. I can't spell chemiosmosis when I'm having a little bit of wine. Osmosis, right? And the final electron acceptor is an organic molecule. It's an organic molecule. Okay, my dog isn't snoring, but she's making a very odd noise right now. I think she is just like wiping her nose on the carpet because it's raining today and she's very wet, and dogs don't like having a wet face for some reason. So if you hear this funny noise, oh, that was her jingling. Come on, girl, come on. She's not listening. If you hear her jingling, that's probably why. Anyhow, fermentation and anaerobic respiration are not the same thing. Come on, girl. Come on. Yeah, you can go up there. She's looking at me like she wants to be up on the couch. Oh, good girl. Good girl. That's right. You stay on the blanket because you're a wet dog. Good girl. Yep. Friday night. This is my exciting Friday night, everybody. It might be your exciting Saturday night. Who knows? But you get to hear me have a little sip of wine, talking biology, dropping a little knowledge, and using my doggy voice. Lucky you. This is the glamorous life, people. The glamorous life of a community college professor. The glamorous life of a student. Here we go. 2020 is interesting. All right, so I wanted to go through that for you so that you were aware of what anaerobic respiration is. It is not the same thing as fermentation, so don't go thinking that it is. Not at all. It's respiration. You still have chemiosmosis. You still have that PMF. It's just that the final electron acceptor is inorganic, and you get way more ATP out of respiration than you do fermentation. Now, there is another option for making ATP. And only certain organisms, certain cells have the privilege of being able to do this. We cannot do this as humans. We're not plants, we're not algae, we're not photosynthetic bacteria. I am talking about photosynthesis. Photosynthesis is the process by which certain organisms are able to harness energy from the sun to make ATP. Using energy in sunlight to make ATP. It is done by organisms that we call photoautotrophs. Photoautotrophs, well, that refers to sunlight, using energy from sun. And autotrophs means that they are able to use an inorganic carbon source. Carbon source. And the one that they use is CO2, for example. CO2 is inorganic. It has carbon, but it does not have hydrogen at all. Okay, so photoautotrophs do photosynthesis. Photosynthesis looks a lot like aerobic respiration with a major caveat. It takes 6 CO2 molecules and 6 water molecules and makes some sugar molecules out of it. C6H12O6 plus 6O2. It spits out some oxygen, and this is what recycles oxygen back into the planet. Those of us who do aerobic respiration, we're putting out a lot of CO2 when we breathe out. We put that into the atmosphere. Plants take up that CO2 and they actually make themselves a sugar molecule from it and also spit out some molecular oxygen. There we go. Okay, so we need a different color. So basically, they are taking CO2, an inorganic carbon source, and they're making a sugar molecule from it. Now think about this for a moment. Is that an oxidation or a reduction? Think about Leo goes Ger. Lose an electron or hydrogen, it's an oxidation. Gain an electron or a hydrogen, it's a reduction. This is a reduction. Carbon is reduced. Carbon dioxide is reduced. Okay, so the last two that we connect here, the H2O forming O2, this is an oxidation. And just like with, so it's kind of the opposite in a way of aerobic respiration. In fact, this is the overall chemical reaction for photosynthesis is the opposite. It's aerobic cellular respiration going backwards, isn't it? So the oxygen thing here, this happens last in aerobic cellular respiration, but oxygen is produced in photosynthesis first. So this oxidation happens first. Whereas the reduction, carbon being fixed and turned into sugar molecules, carbon dioxide being fixed and turned into sugar molecules, this happens last. So this first process here happens first in what we call the light-dependent reactions. Whereas carbon dioxide being fixed and turned into sugars by photosynthetic organisms, plants, algae, photosynthetic bacteria, this happens last in what we call the light independent, I'm going to abbreviate independent, reactions. So there we go. So we've got to look at what we call the light dependent reactions. I am abbreviating the word dependent. I know it looks like the light department. It's not light department, it's light dependent reactions. And the light independent reactions. Got to save myself some writing space here. Save you some too. All right, so we used to call these the light reactions, and the light independent reactions were the dark reactions. But then people used to think that these had to happen in the dark. They don't have to happen in the dark. It's just that they don't require light, whereas the light dependent reactions depend on there being sunlight. So the light dependent reactions happen first. The light independent reactions happen next. Let's focus on the light-dependent reactions for a moment here. These harness energy from sunlight to make ATP. So period, it's all about making ATP. Whereas the light-independent reactions use the ATP from the light-dependent reactions. So they use ATP from the light-dependent reactions to make sugars. To make sugars from CO2. So there we go. So the light-dependent reactions produce ATP. The light-independent reactions use up that ATP to make sugars. And if you're wondering why in the heck would a plant or any photosynthetic organism want to make sugars for itself, think about it. These sugars can be broken down by cellular respiration. Plants don't always have sunlight available to them. There are times when plants need energy and they don't have sunlight around, such as at night, or especially when plants produce their seeds. They pack a lot of sugar either in the embryo around the seeds or within the seed itself, so that when these seeds are planted in the ground, like let's say a pea seed. Peas are really sweet, people. If you've ever eaten peas, they're really sweet. But those pea seeds, the round peas that you eat, those are actually the seed. You plant them in the ground. There's no sunlight under the ground if you bury it in there. Those cells begin to break, the seed begins to break down the sugars that have been packed in there by aerobic cellular respiration. So think of it as plants and algae and photosynthetic bacteria are making their own sugars that they can break down for energy later. It's really cool. They can make their own ATP using energy from sunlight to make sugars that they can break down whenever there's no sunlight around or anytime they need it. So I just wanted to point that out, saying that photosynthetic bacteria and algae and plants do both aerobic cellular respiration and photosynthesis. They don't do just photosynthesis. But hey, we're talking about photosynthesis here, so let's talk about the structures where this happens, and let's focus on eukaryotes, and we can talk about the chloroplast. So the chloroplast is very similar to the mitochondria in that it has an outer membrane and an inner membrane, which is very folded. And you already saw my little sketch of a chloroplast. The inner membrane is highly folded, but they're not folded as in the cristae of the mitochondria. They're stacked like this. Does anybody remember what those stacks are called? These stacks are called thylakoid membranes. They are thylakoid membranes. And within the thylakoid membranes, there are lots of proteins that are embedded in there. And these proteins will form clusters, and they are complexes that we call photosystems. And they contain pigments. Now, if you're wondering, okay, what is a pigment? I think I may have painted with those one time. You'd be correct. A pigment is just a molecule that imparts a color. That's all. If you have ever, taken an art class or heck, you could just walk into an art supply store like Michael's, and you could buy a tube of pigment. You could buy a tube of ultramarine, for example, is a pigment that is a very bright blue, like that. Maybe a little more intense than that color there. And they're just molecules that will absorb, they have a particular color because they absorb light of a particular wavelength. So pigments absorb light of particular, it's the wine talking, people. It's just the wine that's making me not able to spell. No, I'm just kidding. They absorb light of a particular wavelength. In physics and in chemistry, the symbol for wavelength is the Greek letter lambda, and that's what a lambda is. Right there, it looks like a knitting symbol, actually. Lambda means wavelength, and you've probably seen the electromagnetic spectrum at some point. High wavelength will be up here and low here. I don't know why they do it that way, but they do. And let's say, I don't know, chlorophyll, visible light will be like right in the middle. Visible light, and UV light will be here, and like radio waves are up here, I think. Don't quote me on that, but chlorophylls will absorb light over a particular range of wavelength, like maybe over this range. And if there are carotenoids, they'll absorb over maybe that range. And so maybe some other pigments will absorb over those ranges, so that you get these photosystems will absorb light over, this whole range of wavelengths. And again, maybe it's the line talking, but doesn't this kind of look like an emoji? A little bit. Not the meh, but the one with the closed eyes and the closed mouth. I'm losing it, people. It's the second time today I've talked about photosynthesis at length like this. Anyhow, it's fun stuff. What color do I want here? Back to photosystems. Photosystems, they got pigments. Pigments absorb light of a particular wavelength. Actually, and the reason that you can see a particular color is because they're absorbing every color except that color. So for example, our ultramarine here, and no, this is really not what I'd call ultramarine. This color, this pigment is absorbing every color except for this blue, and it's reflecting it back to us. Anywho, what is the big deal about absorbing light of a particular wavelength? What they actually do is they absorb light of a particular wavelength in the form of photons. And I am not a physicist. I'm not even going to pretend that I really, really understand how this works. But a photon is a certain energy unit that exists in light, and it has something to do with this. Do you remember back in a chemistry class, maybe in high school, where you had, or you may have even gotten this in biology, where you talked about, the model of the atom, the Bohr model of the atom, but that electrons can exist in different energy levels, and they drew them as these concentric circles like this, all these different energy levels. And they're like, well, an electron could be in this energy level. And if pigments are designed so that they can absorb light of a particular wavelength, and what it does is it causes them to spit out an electron and bump out an electron to maybe a higher energy level. So it has to do with that. And that, okay, if an electron gets bumped out to this energy level, when it comes back down here, it gives off energy. It has something to do with that. That's the best way I know to explain it. But here's the way it relates to photosynthesis. Actually, if we look back here, if we look back here, and I'll color code these hydrogens here. Right. This part here, this water being split into oxygen, we can't do that. Like if we take H2O, our cells don't have the energy to be able to split it into oxygen and hydrogen. I mean, we could do this if we pulsed some electricity through it. I think I did that in a chemistry lab once. But what happens is that pigments will absorb a photon of light, and they can transfer that energy to the water molecule. So let me draw this a little bit better. Okay, I'm gonna draw this over again. Let me see. H2O. Okay, you water a plant. Plants have pigments that can absorb photons of light, and they can transfer that energy to a water molecule, so that this is what happens. It'll spit out 4 electrons, 4 protons, and some molecular oxygen. Oops, I got to balance that. There we go. Does that look familiar? I sure hope so, because it's the opposite of what happens when oxygen at the end of oxidative phosphorylation binds to the protons and those electrons after these did their little diffusion dance across the inner membrane of the mitochondria. It's the opposite of that. Think of it this way. What could a cell possibly do with these protons? You know what? We could do a proton motive force with these. And that's exactly what happens in the chloroplast and in photosystems. Water is sitting around, and these photosystems are like, hey, I'm going to zap you with some photons of light. And the water splits into oxygen and 4 electrons and 4 protons. Those protons get pumped across the inner membrane of, why did I do that? Get pumped across the inner membrane of the chloroplast in this scenario here. And an electrochemical gradient can be set up, and you get a whole proton motive force. And then ATP can be made when the protons equalize that gradient through ATP synthase. So it's chemiosmosis that is happening there as well. So the whole idea, the whole point of making energy through aerobic respiration and photosynthesis are these. It's all about hydrogens. It's all about getting hydrogen atoms from whatever molecule they're in to split up into electrons and protons here so that you can make a proton motive force. And you can make a ton of ATP from a proton motive force. And that is what plants and photosynthetic bacteria and algae, which are eukaryotes, do through their photosystems. So, photosystems. And again, we are talking the light-dependent reactions. Why am I getting ticked? I swear I turned those notifications off. I have given up. Anywho, let me find a better way to say this. ATP is made by a proton motive force, and you get those protons from the water molecule being split into 4 electrons, 4 protons, and some O2. Oh, there we go. So like that. There are 2 different types of photosystems. There is photosystem 1. Yes, I know I wrote PS1. It looks like PlayStation 1. And photosystem 2. PS2 was my first one. I don't know what we're up to anymore, or what they're even called anymore. Okay, but they both absorb light. So they both have pigments that absorb energy from light. But PS1 actually generates reducing power. And that's in the form of not NADH. They use a molecule called NADPH. But PS2 is what actually splits the water into electrons and protons and oxygen. Now, plants and cyanobacteria and algae have both photosystems. Let's get a different color here. Plants, algae, cyanobacteria have both PS1 and 2. But there are some photosynthetic bacteria that only have one or the other. For example, the green photosynthetic bacteria they have only one photosystem that's similar to PS1. So it's a PS1-like photosystem. Whereas, and I got to change the color for this, the purple bacteria, purple photosynthetic bacteria, can you see it coming? Their photosystem is similar to photosystem 2. Yeah. So that gets us into the light independent reactions. Now, this is where the CO2 is put together to form sugars, that part there. Oh, and by the way, the hydrogens came from water, right? When they're done doing their proton motive force, they'll get fixed and joined with CO2 to form some sugars. But the thing is, it hardly ever happens that 6 are put together to form one glucose molecule. And we'll get to that in just a second. But carbon is put together and it's joined with hydrogen ions and electrons to form sugars through what we call the Calvin cycle. The Calvin cycle, and I love the name of that because it reminds me of Calvin and Hobbes. It has absolutely nothing to do with Calvin and Hobbes. That's too bad. Calvin would like that. The Calvin cycle uses an enzyme called Rubisco to basically put together carbon dioxide molecules with hydrogen. But, gosh, we could spend literally half a semester talking about the Calvin cycle. All I want you to know is that the enzyme Rubisco is used, and it takes 3 turns, turns of the Calvin cycle to make just 1 molecule of glucose. And then it hardly ever happens that you form an entire glucose molecule. Usually, the cell is able to put together just 2 3-carbon molecules are made. So usually just 2 3-carbon molecules are made. What is it? Glyceraldehyde 6-phosphate, I think is the name of it. What is it called? If you go back, and it feeds into glycolysis. You know how glycolysis starts off with C6H12O6 and undergoes a couple of chemical reactions here, and then it is split in half into 2 3-carbon molecules. C3, that's this one. So usually through the Calvin cycle, it's not complete, and they don't get all the way to glucose. Because if they're just going to break it down, why go make it here? They make it here. So it's very interesting. So we kind of lie to you a little bit when you first learn about photosynthesis, that this chemical reaction up here hardly ever happens this way. You hardly ever get one C6H12O6 molecule at all. Doesn't work that way. But this is the balanced equation. But it's really stretching the truth just a little bit. You know, it would be helpful if I actually, what on earth? I don't know why I had this all set up for you beforehand. I don't know why it keeps reverting to some other slide. Anyhow, so photosynthesis, C6H, light reactions, dark reactions, we don't use those terms anymore. They're the light dependent and light independent reactions. Pigments, you just need to know that there are chlorophylls and bacterial chlorophylls. Accessory pigments, I'm not going to be asking you about those, or reaction center versus antenna pigments, so don't worry about that. But you do need to know the two different photosystems. You need to know what goes on in the light dependent reactions. You need to know which photosystem purple and green photosynthetic bacteria have. Okay, I'm not going to ask you to draw anything like this. This is, if we were in person, I would be referring to this a little bit. But you do need to know the difference between photosystem 1 and photosystem 2. Talked about that. Talked about these. Calvin cycle occurs in 3 stages. You do not need to know the total number of ATP and NADH is made. I'm totally fine with that. This is basically a crash course in photosynthesis. What I care about is that you understand that it's about hydrogen. It's about getting hydrogen out of a molecule. In the case of photosynthesis, it's the hydrogens in water. And that those hydrogens are able to be split into their electrons and protons, and those protons are used for PMF, and that's what makes ATP. That's the big deal. Big hairy deal about that. So that gets us into why? Why does it keep I give up. That's like some old chart that I saved a long time ago in Dropbox. But whatever. Chapter 7, Molecular Genetics. I like to call this chapter From Gene to Protein. So we are into Chapter 7, From Gene to Protein. And to get you started on that, when I was an undergraduate, I worked in a microbiology lab part-time while I was going to school, although I don't know why I'm saying part-time, because I worked like 38 hours a week there, so that's not exactly part-time. But I went in through the back door of the building, through the basement, because it was just easier to get in that way. And there was this sidewalk there, and on the sidewalk, it had the letters DNA, and then an arrow to RNA, and then another arrow, and then right in front of the door, it said protein. And I was like, wow, that really kind of says it all. In fact, this little thing right here, this is the theme of chapter 7, although I will add one little thing in that DNA. Sometimes we need to make more of itself, so I'd put a little arrow cycling back there. Sometimes more DNA needs to be made. The DNA is used to make a copy, and that copy is called RNA, and the RNA is used to make a protein. And just as a refresher, the process of the DNA being copied into RNA is called transcription. And the process of the RNA being read and the information in there used to make a protein is called translation. And we will talk about transcription and translation, but first we're going to talk about this little part right here, which is DNA replication. And it looks like it's time for me to plug in my iPad. But I don't have my charger right here, and I am not going to let this lecture fail. So we will find a charger right here. You poor students having to listen to me with my neuroses here. All right. DNA replication is necessary for a cell to be able to divide. So DNA replication means to make a copy of the DNA. If we have a cell here and it needs to divide, it's got a chromosome in there, you can't break the chromosome in half. You got to make 2 copies. You got to make a copy of the chromosome, and then you divvy everything up and the cell can divide that way. So DNA replication is the topic for our Friday evening here. DNA replication, making copies of the DNA. As a refresher for you, you need to remember the DNA is double-stranded. And that these strands are anti-parallel to each other, meaning there's a 5' end here, a 3' end here. Across from every 5' end on one strand, there's a 3' end on the other strand. Across from every 3' end on one strand, there's a 5' end down there. They're complementary, so that means across from every G is a C. Across from every A is a T. So like that. Oops, I need to draw that A upside down, don't I? Oh, that's right. I drew a G upside down. Anti-parallel, complementary. This is all review, and held together by hydrogen bonds. You know that. 3 bonds between a G and a C, 2 between an A and a T. Blah, blah, blah. So there we go, DNA. So when a copy is made, these hydrogen bonds need to be broken and the 2 strands are separated. So DNA replication, both strands are copied. So the strands need to be broken apart. So I'll erase all that stuff there. I'm not getting rid of the G's, A's, T's, and C's. I just want to separate those strands. So both strands are copied, and DNA replication is bidirectional, meaning it happens in both directions. We need a different color here. So this strand on the left will be copied, and the other strand will be copied in the opposite direction. Like that. It is bidirectional. It is also what we call semi-conservative. Semi-conservative means that each new DNA molecule will have 1 original strand and 1 new strand. So that's why I color-coded this for you. What happens when the cell makes a copy of each strand, and I'm just going to move them farther apart here. So here's our original strand in orange. A copy will be made of this one, and these 2 are going to stay together. And this new DNA, new double-stranded chromosome will be in one cell. And this one over here will be copied in the opposite direction, and it'll go into the other cell. So each cell is going to have 1 original strand and 1 newly synthesized strand. That is what we call semi-conservative. So if you look back up here at this little thing that I drew here, each one's going to have a strand of purple and a strand of orange on it, if you think of it that way. So let's see, both strands are copied, and it is bidirectional. It is semi-conservative. And we say that it proceeds from 5' to 3'. meaning the new 5' end is made first. And I was very careful in my drawing to do it that way. Like, let's look over here. Let's look here. Notice that I started my arrow down here, right? So what number do I put here? It's across from the 3' one over here, so it's going to be a 5'. This one over here on the right, that's going to be a 5' end because it's across from this 3' end. I made sure that I started my arrow there and I have it pointing in the opposite direction. I was careful to do that. Even with a little glass of wine, I'm like, shoot, I hope I haven't clouded my judgment here. Spoiler alert, I'm really not drinking wine here. I would like to be, but I am just drinking a glass of ice water because that's how I roll and that's how I party on a Friday night. So let's talk about DNA replication. We've already said it's bidirectional, it is semi-conservative, and it proceeds from 5' to 3', but let's look at the nuts and bolts of how this works. There are several different enzymes that you need for DNA replication. Needed for DNA replication. First up is that you need an enzyme that unwinds the DNA. Okay, DNA is not all flat like this all over the place. I keep forgetting that I have to circle what I'm pointing at. You can't see me pointing from here. But DNA exists as a helix, right? So it's all wound up like that, two-strand, and this is all two-stranded in here. Looks like the topping on a Hostess cupcake, but it's supposed to be DNA here. But something's got to unwind this. And the enzyme that does the unwinding is called helicase. Helicase. So helic meaning helix. So it's an enzyme that unwinds the helix. So helicase binds to an area of the DNA called the origin of replication, or the ori. And it's about like a 250 base pair, 250 pairs of those G's, A's, T's, and C's. And helicase will recognize a specific order of those nucleotides and bind to them and begin to unwind the DNA there. So helicase binds to the origin of replication and unwinds the DNA. And it essentially opens it up. So what helicase does is, if we've got this helix of DNA here, it's going to open it up to, it will recognize, like, if this is the origin of replication somewhere in this area here, it'll open it up right there. So we'll have something that looks like that. So I'm separating these far out so that we can see that helicase is right there. It's almost like helicase is a zipper tab. Like this is a little zipper tab right here. That's helicase. And helicase is unzipping. You could think of it as unzipping the DNA. And let's see if we need a 3' end and a 5' end right there. So that's what helicase does. It unwinds the DNA. Now here you're going to have, G, A, T, T, C, A right there. And so on the other strand, there'll be the C, the T that went with that A right there. Go. A, G, T, something like that. So it opens it up, and these hydrogen bonds that held the DNA together are now broken. As helicase unwinds the DNA, that gives a new enzyme room to come in. And that new enzyme is called DNA polymerase. DNA polymerase is an enzyme that adds complementary nucleotides to one of the strand. So DNA polymerase, it's an enzyme that polymerizes or makes a new piece of DNA. Every organism is going to have its own DNA polymerase, its own special type of one. So it's really, DNA polymerase is a class of enzymes that are similar, but like the DNA polymerase that human cells have is going to be different from the DNA polymerase that E. coli has, for example. So they're all going to be a little different, but I'm just calling it DNA polymerase for right now. Okay, so DNA polymerase will also attach to the origin of replication. Attaches to an origin of replication. And it begins adding complementary nucleotides to one of the strands. And adds complementary nucleotides to one of the strands. I need a little more room here. Beginning with, and this is important, and so we'll underline it when we're done with this, beginning with the new 5' and first. Beginning with the new 5' and first. Way back here, I said it's bidirectional and it proceeds from 5' to 3'. This is what I'm talking about. It makes the new 5' end first. So let's see, we need a different color here. Let's go with orange. So DNA polymerase. Will be in orange. So DNA polymerase has the choice of two strands here. It could add, is it going to be able to add complementary nucleotides? Like if we look at these two ends here, here and here, is it going to be able to add a complementary nucleotide to this T first or to this A up here first? Remembering that it has to add the new 5' end first. Can't emphasize that enough. Well, let's take a look what would happen if we add it to the T down here. It'll be like, oh, I see a T here, I'll add an A, but what number do I have to put there? That's a 5' end over here, so I gotta have to put a 3' end here, and DNA polymerase can't work in that direction. Think of it as like zipper teeth. You know how zipper teeth only work in one direction? On certain zippers. They lock in a certain way like that. Maybe that's not such a great analogy, but take home message. DNA polymerase only works in one direction, and its direction is 5' to 3'. So it's got to come up here, and it can add a T there. And because this is the 3' end over here, it has a 5' end right there. So it adds a T, and then it goes to the next one, and it adds a G, and then it adds an A, and another A, and I'm running out of space here, and then a T. Can you see how that happens like that? Then it's going to make it to this area here where helicase was. This is called the replication fork. The replication fork. Think of it as a fork in the road, that sort of thing. Well, once DNA polymerase comes down here, helicase is going to have to unwind a little bit. It'll have to unzip a little bit more to make room for it, won't it? But that's not the only thing that happens. And to do that, to show you what happens next, we need another color here. Let's go with green. Why not? Okay, this strand down here's got to be copied too, right? But and in the cell, there's not going to be one molecule of DNA polymerase doing all this work. There are going to be a bunch of molecules of the same thing. There will be several different molecules of the same DNA polymerase. But it can't start here and add an A here because that's got to be a 3' end right there. This actually, I'm just going to erase this here. To get this strand down here copied, DNA polymerase needs to start down here, doesn't it? And it's going to add an A here because that'll be the 5' end. And I know this is looking like a hot mess here because I can't write teeny tiny here. It'll see this A and add a T. It'll see this A and add another T. It'll see this G and add a C. It'll see this T and add an A, etc., because its 5' end is down there. So it's bidirectional in that this green strand is moving this way and the orange one is moving towards the replication fork. Okay, so as this one, the orange one, and I'll just draw this here, when this DNA polymerase molecule makes it to the replication fork, helicase is like, heck, I better unzip a little bit more. So it will unzip some more. So to show you how that works, how am I going to do this? We're going to erase a little bit. Oh no, I can't do that. It'll erase the whole darn thing, won't it? Ha, actually it didn't. How about that? So I'll go back to purple here. And let's say it'll unwind things even more. Oh God, I realize, what if I did white? That would help things along. Like that. Okay, so now helicase is down here, the zipper tab. There we go. My dog is snoring. So I'll fill in with some nucleotides here. And I've got to get this green one out of the way here. All right, just a little bit. I just took that 5' out. So let's go back to the orange one. Man, this is so much easier to do when you have a big giant whiteboard to write on. But this one where it saw an A and added a T, saw a C and added a G, a T and added an A, another T and added an A, an A added a T. Now it has room that it's unzipped, and so it'll add a C here. I'm just going to draw a line showing that it's moving smoothly in that direction right there. I didn't give myself enough room here, so I'm going to draw this over again, okay? So hang in here with me. Let's say helicase has unwound a lot now. Let's see, that was the 3' end, this is the 5' end. And DNA polymerase started adding nucleotides in this direction because it was the 5' end first. But when the origin of replication was right up here, the green one back here had to start here, where that original origin of replication was. So it started there, and it's moving away from the replication fork because it has to add the 5' new nucleotide first. All right. Can you see that this orange one up here can just keep smoothly heading in the direction of the replication fork? But what about the green one? At this strand down here, what about all this stuff? What's got to happen there is that DNA polymerase can't move backwards, can't do that, because it can only add the 5' nucleotide first, so it has to start over down here and move until it bumps into the last spot where the replication fork was. So you get a little gap here, don't you? So this little gap, that represents where that first replication fork was way back here. So this is the spot of the original replication fork. So you're going to have a little fragment down here. All right, so helicase is like, well, crap, I got to unwind even more. So we're going to have something that looks like this now. Three prime, 5 prime. The orange strand started here with the 5 prime end, and it keeps adding complementary nucleotides, smoothly heading toward the replication fork. But the one down here is going to be fragmented like that, because it has to start over at all the spots where the new replication fork was. Like, it has to start right down here, moving this way. So this smooth strand up here in orange we call the leading strand. And it's because DNA polymerase can start adding to the old 3' end because it has to add the new 5' end first. So it's heading towards the replication fork. The one in green down here is called the lagging strand. And it lags behind, and it's all fragmented because it has to start at each replication fork and move away from the replication fork. So the new DNA piece begins at each new replication fork and moves away from the replication fork. And so it's fragmented. And these fragments are called Okazaki fragments. I suppose someone whose name is Okazaki figured all this stuff out. And in science, if you figure it out, you often get to name it for yourself. So name it after yourself. So there you go. But we got a problem here. DNA does not like having these little gaps here because, you know, this one's going to have a 3' end hanging off of it here, here, and here. And there's this little gap in there. So, oops. There we go. So we need yet another enzyme to fill in these gaps and to join from here to here, like that. And the enzyme that fills in the gaps is called DNA ligase. It ligates the Okazaki fragments to each other. Fills in the gaps and see, it fills in the gaps and joins the Okazaki fragments to each other. So there we go. So that's the gist of DNA replication. You need to know how this leading strand and this lagging strand develop. And that's all because DNA polymerase has got to add the new 5' end first. That's basically the gist of it, and we will talk just a teeny bit more about this next time. I hope wherever you are having a delightful day, and I will see you in class. Take it easy.